Good day, everyone. I am Mr. Jericho Retha from BS Ed 3 Science, and today I am going to discuss about the cell signaling and signal transduction communication between cells. So let us start. These are the 8 subtopics that we will encounter and discuss later. Most cells in a plant or animals are specialized to carry out one or more specific function. Many biological processes require various cells to work together and to coordinate their activities. To make this possible, cells have to communicate with each other, which is accomplished by a process called cell signaling. Cell signaling make it possible for cells to respond in an appropriate manner to a specific environmental stimulus. Cell signaling affects virtually every aspect of cell structure and function, which is one of the primary reasons we were discussing about this chapter. Here are the basic elements of cell signaling system. Cells usually communicate with each other through extracellular messenger molecules. Extracellular messenger can travel a short distance and stimulate cells that are in close proximity to the origin of the message, or they can travel throughout the body, potentially stimulating cells that are far away from the source. You can see at the pictures are the three types of intercellular signaling: the autocrine, paracrine, and endocrine. In the case of autocrine signaling, the cell that is producing the messenger expresses receptor on its surface that can respond to that messenger. Consequently, cells releasing the message will stimulate themselves or inhibit. During paracrine signaling messenger, molecules travel only short distances through the extracellular space to cells that are in close proximity to the cell that is generating the message. Paracrine messenger molecules are usually limited in their ability to travel around the body because they are inherently unstable, or they are degraded by enzymes, or they bind to the extracellular matrix. Finally, during endocrine signaling, messenger molecules reach their target cells via passage through the bloodstream. Endocrine messengers are also called hormones, and they typically act on target cells located at distant sites in the body. Another. The picture you are seeing now is an overview of cellular signaling pathways. So, here are the steps in cellular signaling pathways. First, cell signaling is initiated with the release of messenger molecule by a cell that is engaged in sending messages to other cells in the body. Second, cells can only respond to a particular extracellular message if they express receptor that is specifically recognize and bind that messenger molecule. Ligand is a molecule that binds to the receptor. Step 3. Extracellular messenger molecule binds to a receptor at the outer surface of the responding cell. This interaction induces a conformational change in the receptor that causes the signal to be relayed across the membrane to the receptor's cytoplasmic domain. Step 4. One type of receptor transmits a signal from its cytoplasmic domain to a nerve enzyme and generates a second messenger. Step 6. Whether the signal is transmitted by a second messenger or by protein recruitment, the outcomes is similar. A protein that is positioned at the top of an intercellular signaling pathway is activated. Step 7. Signaling pathways are the information superhighways of the cell. Each signaling pathways consists of a series of distinct proteins that operate in sequence. Step 8. Signal transmitted along such signaling pathways ultimately reach target proteins. And when it reaches the target proteins, the cellular processes may begin or the signal transduction may occur. These overall processes in which information carried by extracellular messenger molecules is translated into changes that occur inside a cell is referred to as signal transduction. This picture is an image of a signal transduction pathway consisting of protein kinases and protein phosphates whose catalytic actions change the conformation and thus the activities of the protein they modify. Another picture you are seeing now is an image of a comparison in the frequency of tyrosine phosphorylation in two different types of breast cancer cells. The panel on the left side of the figure shows the frequency of phosphotyrosine residues in certain proteins in a triple negative breast cancer cell lines. These triple negative cells do not express three major molecular signatures of many breast cancer cells: the estrogen receptor, the progesterone receptor, and the growth factor receptor or HER2. The frequency of the phosphotyrosine residue in breast cancer cells that express these three signature proteins is shown in the panel on the right. The frequency of the phosphotyrosine residue in a given protein in a given cell line is indicated by the intensity of the red shading of the box. The names of the cell lines tested are given along the top of the figure. It is evident that the triple negative cells have a much greater level of tyrosine phosphorylation than the other breast cancer cells. This may correlate with the loss of a particular protein tyrosine phosphatase activity in many of the triple negative cancers. Moving on to our next subtopic, which deals about on a survey of extracellular messenger and their receptors. Okay, a large variety of molecules can function as extracellular carriers of information. This includes first, amino acid and amino acid derivatives. Example, glutamate, glycine, acetylcholine, epinephrine, dopamine, and thyroid hormone. These molecules act as a neurotransmitter in the hormones. Second, gases such as nitric oxide and carbon oxide. Third, steroids, which are derived from the cholesterol. Steroid hormones regulate sexual differentiation, pregnancy, carbohydrate metabolism, and excretion of sodium and potassium ions. Fourth is eicosanoids, which are nonpolar molecules containing 20 carbons that are derived from a fatty acid named arachidonic acid. These eicosanoids regulate a variety of processes including pain, inflammation, blood pressure, and blood clotting. Several over-the-counter drugs that are used to treat headaches and inflammation inhibit eicosanoid synthesis. Lastly, a wide variety of polypeptides and proteins. Some of these are present as transmembrane proteins on the surface of an interacting cell. Others are part of the extracellular matrix. And finally, a large number of proteins are excreted into the extracellular environment where they are involved in regulating processes such as cell division, differentiation, the immune response, or cell death and cell survival. Extracellular signaling molecules are usually recognized by specific receptors that are present on the surface of the responding cell. Receptors bind their signaling molecules with high affinity and translate this interaction at the outer surface of the cell into changes that takes place on the inside of the cell. The receptors that have evolved to mediate signal transduction are indicated below. First, the G protein coupled receptors or the GPCRs is a huge family of receptors that contain 7 transmembrane alpha helices. Second, receptor protein tyrosine kinases or RTKs that represent as a second class of receptor that have evolved to translate the presence of extracellular messenger molecules into changes inside the cell. Third, the ligand-gated channels that represent as a third class of cell surface receptor that bind to extracellular ligands. The ability of this protein to conduct a flow of ions across the plasma membrane is regulated directly by ligand binding. Fourth, steroid hormone receptor that function as a ligand-regulated transcription factors. And finally, there are a number of other types of receptors that act by unique mechanism. Some of these receptors, for example, is the B and T cell receptor that are involved in the response of foreign antigens associated with known signaling molecules such as cytoplasmic protein tyrosine kinases. Another subtopic is the G protein coupled receptor and their second messenger. So first, we will define what is GPCRs. G protein coupled receptor or GPCR are so named because they interact with G proteins. The members of the GPCR superfamily are also referred to as 7 transmembrane receptor because they contain 7 transmembrane helices. Thousands of different GPCRs have been identified in organisms ranging from yeast to flowering plants and mammals that together regulate an extraordinary spectrum of cellular processes. And in fact, GPCRs constitute the single largest superfamily of proteins encoded by animal genomes. Included among the natural ligands that bind to GPCRs are a diverse array of hormones from both plant and animals, and the neurotransmitter, opium derivatives, chemoattractants, or the molecules that attract phagocytic cells of the immune system, odorants and testants, or the molecules detected by olfactory and gustatory receptor, eliciting the senses of smell and taste, as well as the photons. The image we can see on the slide is the membrane-bound machinery for transducing signal by means of a 7 transmembrane receptor in a heterotrimeric G protein. Receptor of this type, including those that bind epinephrine and glucagon, contain 7 transmembrane spanning helices. When bound to their ligand, the receptor interacts with a trimeric G protein, which activates an effector such as adenylyl acyclase. As indicated in the figure, the A and G subunits of the G protein are linked to the membrane by lipid groups that are embedded in the lipid bilayer. G protein coupled receptor normally have the following topology. Their amino terminus is present on the outside of the cell. The 7 alpha helix that traverse the plasma membrane are connected by loops of varying length, and the carboxyl terminus is present on the inside of the cell. There are also 3 loops present on the outside of the cells that together form the ligand binding packet, whose structure varies among different GPCRs. There are also three loops present on the cytoplasmic side of the plasma membrane that provide binding sites for intracellular signaling proteins. To continue, let us discuss the signal transduction by G-protein coupled receptor. G-proteins, heterotrimeric G-proteins, were discovered, purified, and characterized by Martin Rodville and his colleagues at the National Institute of Health and Alfred Gilman and colleagues at the University of Virginia. These proteins are referred to as G-protein because they bind guanine nucleotides, either GTP or GDP. They are described as heterotrimeric because all of them consist of three different polypeptide subunits called A, B, and G. This property distinguishes them from small monomeric G proteins such as RAS, which are discussed later in this chapter. Heterotrimeric G proteins are held at the plasma membrane by lipid chains that are covalently attached to the A and G subunits. Heterotrimeric G proteins come in four flavors. First, GS, GQ, GI, and G12/13. This classification is based on the GA subunits and the effector to which they couple. The particular response elicited by an activated GPCR depends on the type of G protein with which it interacts. Although some GPCRs can interact with different G proteins and trigger more than one physiologic response, GS family members couple receptors to adenylyl cyclase. Next, termination of the response. We have seen that the ligand binding results in receptor activation. The activated receptor turn on G proteins and G proteins turn on effectors. To prevent overstimulation, receptors have to be blocked from continuing to activate G proteins. To regain sensitivity to future stimuli, the receptor, the G protein, and the effector must all be returned to their inactive state. Desensitization. It is the process that blocks active receptors from turning on additional G proteins takes place in two steps. First step, the cytoplasmic domain of the activated GPCR is phosphorylated by a specific type of kinase called G-protein coupled receptor kinase or GRK. These GRKs form a small family of serine-threonine protein kinases that specifically recognize activated GPCR. Second step, the phosphorylation of the GPCR set the stage, which is the binding of proteins called aristines. Aristine form a small family of protein that bind to GPCRs and complete for binding with heterotrimeric G protein. As a consequence, aristine binding prevents the further activation of additional G protein. This action is termed desensitization because the cell stops responding to the stimulus while that stimulus is still acting on the outer surface of the cell. Desensitization is one of the mechanisms that allows a cell to respond to a change in its environment rather than continuing to fire endlessly in the presence of an unchanging environment. The importance of desensitization is illustrated by the observation that mutations that interfere with phosphorylation of rhodopsin by GRK leads to the death of the photoreceptor cells in the retina. This type of retinal cell death is thought to be one of the causes of blindness resulting from the disease retinitis pigmentosa. Another requirement in the termination of the response in cell signaling is the arrestin. Arrestin can be described as protein hubs in that they are capable of binding to a variety of different proteins involved in different intracellular processes. Arrestin molecules are also capable of binding to AP2 adapter molecules that are situated in clarithin-coated pits. The clear example is the image I'll shown in the next slide. This image shows an arrestin-mediated internalization of GPCRs. Arrestin bonds are internalized when they are trapped in clarithin-coated pits which bud into the cytoplasm. Clarithin-coated buds are transformed into clarithin-coated vesicles which deliver their contents, including the GPCRs, to endosomes. When present in the endosomes, arrestin serves as scaffolds for the assembly of signaling complexes, including those that activate the MAPK cascades and the transcription factor ERK. Alternatively, the GPCRs can be delivered by lysosomes where they are degraded, or they can be returned to the plasma membrane in a recycling endosomes where they can interact with new extracellular ligands. We will discuss about the effects of certain mutations on the function of G protein coupled receptors. Since GPCRs represents the largest family of genes encoded by the human genome, their importance in human biology is reflected by the fact that over one-third of all prescription drugs act as a ligand that binds to this huge superfamily of receptor. A number of inherited disorders have been traced to the plex in heterotrimeric G protein. As noted in Table 1, mutation in genes that encodes the subunits of heterotrimeric G proteins can also lead to inherited disorder. This is illustrated by a report on two male patients suffering from a rare combination of endocrine disorder, precocious puberty, and hypoparathyroidism. Both patients were found to contain a single amino acid substitution in one of the GA isoforms. The alteration in amino acid sequence caused the two effects on the mutant G protein. At temperatures below normal body temperature, the mutant G protein remained in the active state even in the absence of a bound ligand. Because G proteins are so important to the normal physiology of multicellular organism, they provide excellent targets for bacterial pathogens. For example, cholera toxins exerts its effects by modifying GA subunits and inhibiting their GT phase activity in the cell of the intestinal epithelium. As a result, adenyl cyclase molecules remain in an activated mode, churning out CAMP, which causes the epithelial cells to secrete large volumes of fluid into the intestinal lumen. The loss of water associated with this inappropriate response often leads to death due to dehydration. And now, we will discuss about the discovery of cyclic AMP or CAMP, a prototypical second messenger. Cyclic AMP is a second messenger that is capable of diffusing to other sites within the cell. The synthesis of cyclic AMP follows the binding of a first messenger, a hormone or other ligand, to a receptor at the outer surface of the cell. To clearly understand about cyclic AMP, a second messenger, here is a photograph showing the localized formation of CAMP in a live cell in response to the addition of an extracellular messenger molecule. This series of photographs shows a sensory nerve cell from the C hair or aplasia. The concentration of pre-CAMP is indicated by the color. Blue represents a low CAMP concentration, yellow an intermediate concentration, and red a high concentration. The left image shows the intracellular CAMP level in the unstimulated neuron. In the next three images, show the effect of stimulation by the neurotransmitter serotonin at the times indicated. Notice that the CAMP levels drop by 109S despite the continued presence of the neurotransmitter. The CAMP level was determined indirectly in this experiment by microinjection of a fluorescently labeled CAMP dependent protein kinases labeled with both fluorescein and rhodamine on different subunits. Energy transfer between the subunits provide a measure of CAMP concentration. Now, let us discuss about the phosphatidylinoacetyl-derived second messengers. The phospholipids of cell membranes were considered strictly as structural molecules that made membranes cohesive and impermeable to aqueous solids. Our appreciation of phospholipids has increased with the realization that these molecules form the precursor of a number of second messengers. Phospholipids of cell membranes are converted into second messengers by a variety of enzymes that regulate in response to extracellular signals. These enzymes include phospholipase, phospholipid kinases, and phospholipid phosphatase. Phospholipase are enzymes that hydrolyze specific ester bonds that connect the different building blocks that makes up a phospholipid molecule. This picture is about the phospholipid-based second messengers. The first image is the structure of a generalized phospholipids. Phospholipids are subject to attack by four types of phospholipase that cleave the molecule at the indicated sites. Of these enzymes, we will focus on PLC, which splits the phosphorylated head group from the diacylglycerol. And the other photograph is a model showing the interaction between a portion of a PLC enzyme molecule containing a pH domain that binds to the phosphorylated inositol ring of a phosphoinositide. This interaction holds the enzyme to the inner surface of the plasma membrane and may alter its enzymatic activity. This slide will talk about the phosphatidylinoacetyl phosphorylation. When the neurotransmitter acetylcholine binds to the surface of a smooth muscle cell within the wall of the stomach, the muscle cell is stimulated to contract. When a foreign antigen binds to the surface of a muscle, the cell is stimulated to secrete histamine, a substance that can trigger the symptoms of an allergy attack. Both of these responses, one leading to contraction and the other to secretion, are triggered by the same second messenger, a substance derived from the compound phosphatidylinositol, a minor component of most cellular membranes. The fourth subtopic that tasked to discuss by Ming is the protein tyrosine phosphorylation as a mechanism for signal transduction. First, let us define protein tyrosine kinase. It is an enzyme that phosphorylates a specific tyrosine residue on protein substrate. And here comes the protein tyrosine phosphorylation. It is a mechanism for signal transduction that appeared with the evolution of multicellular organisms. Over 90 different protein tyrosine kinases are encoded by the human genome. These kinases are involved in the regulation of growth, division, differentiation, survival, attachment to the extracellular matrix, and migration of cells. Protein tyrosine kinases can be divided into two groups. Receptor protein tyrosine kinases or RTKs, which are integral membrane proteins that contain a single transmembrane helix and an extracellular ligand binding domain. Another is non-receptor or cytoplasmic protein tyrosine kinases. For your information, the human genome encodes nearly 60 RTKs and 32 non-receptor TKs. These RTKs are activated directly by extracellular growth and differentiation factors such as epidermal growth factor or EGP and platelet-derived growth factor or PDGP or biometabolic regulators such as insulin. Non-receptor protein tyrosine kinases are regulated indirectly by extracellular signal and they control processes as diverse as the immune system, cell adhesion, and neuronal cell migration. And now, we will talk about the receptor dimerization. Structural biologists have yet to solve the three-dimensional structure of an entire RTK. It is widely accepted on the basis of other techniques that ligand binding result in the dimerization of the extracellular ligand binding domains of a pair of receptor. Two mechanisms for receptor dimerization have been recognized. First is ligand-mediated dimerization, and second is receptor-mediated dimerization. This image is a step in the activation of a receptor protein tyrosine kinase or RTKs. The first picture on the left side is ligand-mediated dimerization. In the non-activated state, the receptors are present in the membrane as monomers. Binding of a bivalent ligand leads directly to dimerization of the receptor in the activation of its kinase activity, causing it to add phosphate groups to the cytoplasmic domain of the other receptor subunit. The newly formed phosphorotyrosine residue of the receptor serves as a binding site for target protein containing either SH2 or PTB domains. The target proteins become activated as a result of their interaction with the receptor. In the image on the right side showing receptor-mediated dimerization. The sequence of events is similar to those in Part A, except that the ligand is monovalent and consequently, separate ligand molecules bind to each other of the inactive monomers. Binding of each ligand induces a conformational change in the receptor that creates a dimerization interface. The ligand-bound monomers interact through this interface to become an active dimer. Next slide is about the Ras-MAP kinase pathway. Retroviruses are small viruses that carry their genetic information in the form of RNA. Some of these viruses contain genes called oncogenes that enable them to transform normal cells into tumor cells. RAS was originally described as the product of a retroviral oncogene and only later determined to be derived from its mammalian host. It was subsequently discovered that approximately 30% of all human cancers contain mutant versions of RAS genes. At this point, it is important to note that the that RAS proteins are part of superfamily of more than 150 small monomeric G proteins including the RABs, SARL, and RAND. These proteins are involved in the regulation of numerous processes including cell division, differentiation, gene expression, cytoskeletal organization, vesicle trafficking, and nucleocytoplasmic transport. The principles discussed in connection with RAS apply to many members of the small G protein superfamily. RAS is a small GT phase that is anchored at the inner surface of the plasma membrane by a covalently attached lipid group that is embedded in the inner leaflet of the bilayer. Along this RAS map kinase pathway, mutation occurs. Mutation in one of the human RAS genes that lead to tumor formation prevent the protein from hydrolyzing the bound GTP back to the GDP form. As a result, the mutant version of RAS remain in the on position, sending a continuous message downstream along the signaling pathway and keeping the cell in the proliferative mode. The cycling of monomeric G proteins, such as RAS, between active and inactive state is aided by accessory proteins that bind to the G protein and regulate its activity. These accessory proteins include GTPase activating proteins or GAPs, guanine nucleotide exchange factor or GEFs, and guanine nucleotide dissociation inhibitors or GDIs. The activity and localization of these various accessory proteins are tightly regulated by other proteins, which those regulate the state of the G protein. We will now move to the fifth subtopic, which dealing on the role of calcium as an intracellular messenger. Calcium ions play a significant role in a remarkable variety of cellular activities, including muscle contraction, immune responses, cell division, secretion, fertilization, synaptic transmission, metabolism, transcription, cell movement, and cell death. In each of these cases, an extracellular message is received at the cell surface and leads to a dramatic increase in concentration of calcium ions within the cytosol. The concentration of calcium ion in a particular cellular compartment is controlled by the regulated activity of cytosolic pumps, cytosolic exchangers, and cytosolic ions channels that is located within the membranes that surround the compartment. The concentration of cytosolic ions in the cytosol of a resting cell is maintained at very low levels, typically about 10 raised to 27 m. In contrast, the concentration of this ion in the extracellular space or within the lumen of the ER or a plant cell vacuole is typically 10,000 times higher than the cytosol. The cytosolic calcium level is kept very low because cytosolic ion channels in both the plasma membrane and endoplasmic reticulum membranes are normally kept closed, making these membranes highly impermeable to these ions. And another energy-driven cytosolic transport system of the plasma and ER membranes pump calcium out of the cytosol. Abnormal elevation of cytosolic concentration can occur in brain cells following a stroke can lead to massive cell death. The picture depicted is an experiment demonstration of localized release of intracellular cytosolic within a single dendrite of a neuron. In the micrograph shown here, which pictures an immensely complex Purkinje cell or neuron of the cerebellum, calcium ions have been released locally within a small portion of the complex dendritic tree. Calcium releases from the endoplasmic reticulum, showed in red color, was induced in the dendrite following the local production of IP3, which followed the repetitive activation of a nearby synapse. The sites of a release of cytosolic ions are revealed by fluorescence from a fluorescent calcium indicator that was loaded into the cell prior to stimulation of the cell. This slide deals with the IP3 and voltage-gated cytosolic channel. We have described in previous pages the two major types of signaling receptor, which are GPCRs and RTKs. Interaction of an extracellular messenger molecule with a GPCR can lead to the activation of the enzyme phospholipase, which splits the phosphoinositide PIP2 to release the molecule IP3, which opens calcium channel in the endoplasmic reticulum membrane, leading to a rise in cytosolic. Extracellular messenger that signals through RTKs can trigger a similar response. The primary difference is that RTKs activate members of the phospholipase CG subfamily, which possess an SH2 domains that allows them to bind to the activated phosphorylated RTKs. The photograph you can see on the screen is calcium-induced calcium release, as it occurs in a cardiac muscle cell. A depolarization in membrane voltage causes the opening of voltage-gated calcium channels in the plasma membrane, allowing entry of a small amount of cytosolic into the cytosol. The calcium ion binds to ryanodine receptor in the SER membrane, leading to release of stored cytosolic in the cytosol, which trigger the cell construction. The calcium ions are subsequently removed from the cytosol by the action of cytosolic pumps located in the membrane of SER, and a secondary transport system in the plasma membrane which lead to relaxation. This cycle is repeated after each heartbeat. Moving on to a calcium binding proteins. Unlike CAMP, whose action is usually mediated by stimulation of protein kinase, calcium can affect a number of different types of cellular effectors, including protein kinases. Depending on the cell type, calcium ions can activate or inhibit various enzymes in transport systems, change the ionic permeability of membranes, induce membrane fusion, or alter cytoskeletal structure induction. Calcium does not bring about these responses by itself but act as a conjunction with a number of calcium-binding proteins. The best studied calcium-binding protein is calmodulin, which participates in many signaling pathways. Calmodulin is found universally in plants, animals, and eukaryotic microorganisms, and it has virtually the same amino acid sequence from one end of the eukaryotic spectrum to the other. Each molecule of calmodulin contains 4 binding sites for calcium. Calmodulin does not have sufficient affinity for calcium to bind the ion in a non-stimulated cell. This picture is about calmodulin, a ribbon diagram of calmodulin with 4 bound calcium ions, or white spear. Binding of these calcium ions change the conformation of calmodulin, exposing a hydrophobic surface that promotes interaction of calcium with a large number of target proteins. This slide will focus on the regulating calcium concentration in plant cell. Calcium ion, as an acting in conjunction with calmodulin, are important intracellular messenger in plant cell. The levels of cytosolic calcium change dramatically within certain plant cells in response to a variety of stimuli, including changes in light, pressure, gravity, and the concentration of plant hormones such as abscisic acid. The concentration of calcium in the cytosol of a resting plant cell is kept very low by the action of transport proteins situated in the plasma membrane and vacuolar membrane or tonoplast. Here is the photograph of a simplified model of the role of calcium in guard cell closure. In the first image on the left side of the screen is the photograph of stomatal pores, each flanked by a pair of guard cells. The stomata are kept open as turgor pressure is kept high within the guard cell, causing them to bulge outward as seen here in the picture. And in the right side of the screen is an image showing one of the factors controlling stomatal pore size is the hormone abscisic acid or ABA. Let us move on the sixth subtopic entitled Convergence, Divergence, and Crosstalk among different signaling pathways. The signaling pathways described above in the various figure depict linear pathways leading directly from a receptor at the cell surface to an end information through a cell, not unlike the way their central changes produce a net outflow of K ions and anions and a resulting decrease in turgor pressure target. In actual fact, signaling pathways in the cell are much more complex. For example, signals from a variety of unrelated receptors, each binding to its own ligand, can converge to activate as common effectors such as RAS and RAP. Another, signals from the same ligand such as EGF or insulin can diverge to activate a variety of different effectors and pathways leading to diverse cellular responses. Lastly, signal can be passed back and forth between different pathways, a phenomenon known as crosstalk. To be able to understand easily, here is an example of convergence, divergence, and crosstalk among various signal transduction pathways. These drawings show the outlines of signal transduction pathways initiated by receptors that act by means of both heterotrimeric G proteins and receptor protein tyrosine kinases. The two are seen to converge by the activation of different phospholipase C isoforms, both of which lead to the production of the same second messenger, which are IP3 and DAG. Activation of the RTKs by either PDGP or EGF leads to the transmission of signals along three different pathways. An example of divergence. Crosstalk between the two types of pathways is illustrated by calcium ions, which are released from the SER by action of IP3 and can then act on various proteins including protein kinase C. That picture or that slide show an example of convergence, divergence, and crosstalk among signaling pathways. This image shows that a signal transmitted from a G protein-coupled receptor, an integrin, and a receptor tyrosine kinases all converge on RAS and are then transmitted along the MAP kinase cascade. The recruitment of the GRV2 complex result in the activation of RAS and transmission of signals down the MAP kinase pathway. And as a result of this convergence, signals from diverse receptors can lead to the transcription and translation of a similar set of growth-promoting genes in each target cell. Another photograph is an example of crosstalk between two major signaling pathways. Cyclic AMP acts in some cells by means of the CAMP-dependent kinase pKa to block the transmission of signals from RAS to RAP, which inhibits the activation of MAP kinase cascade. In addition, both pKa and the kinase of the MAP kinase cascade phosphorylate the transcription factor CRIB on the same serine residue, activating the transcription factor and allowing it to bind to a specific site on the DNA. Moving on the next subtopic about the role of nitric oxide as an intercellular messenger. During the 1980s, a new type of messenger was discovered that was neither an organic compound such as cyclic AMP nor an ion such as calcium ion. It was an inorganic gas called nitric oxide. So, nitric oxide is unusual because it acts both as an extracellular messenger, mediating intracellular communication, and as a second messenger, acting within the cell in which it's generated. Nitric oxide is formed from the amino acid L-arginine in a reaction that requires oxygen and NADPH, in that is catalyzed by the enzyme nitric oxide synthase or NOS. Since its discovery, it has become evident that nitric oxide is involved in a myriad of biological processes including anticoagulation, neurotransmission, smooth muscle relaxation, and visual perception. As with many other biological phenomena, the discovery that nitric oxide function as a messenger molecule began with an accidental observation. It had been known for many years that acetylcholine acts in the body to relax the smooth muscle cells of blood vessels, but the response could not be duplicated in vitro. When portion of a major blood vessels such as the aorta were incubated in physiologic concentration of acetylcholine in vitro, the preparation usually showed little or no response. Another image is showing a signal transduction pathway that operates by means of nitric oxide in cyclic GMP that leads to the dilation of blood vessels. And now, we will discuss the steps in binding of acetylcholine to the outer surface of an endothelial cell. Signal arise in cytosolic calcium ions concentration that activates nitric oxide synthase. The nitric oxide formed in the endothelial cell diffuses across the plasma membrane and into the adjacent smooth muscle cells, where it binds and stimulates guanylyl cyclase, the enzyme that synthesizes cyclic GMP, which is an important second messenger similar in structure of CMP. Cyclic GMP binds to cyclic GMP dependent protein kinase or a PKJ, which phosphorylate specific substrates causing relaxation of the muscle cell and dilation of the blood vessel. Nitric oxide also serves as an activator of guanylyl cyclase. The discovery that nitric oxide acts as an activator of guanylyl cyclase was made in the late 1970s by Farid Murad and his colleague at the University of Virginia. Murad was working with acide or N3, a potent inhibitor of electron transport, and a chance to discover that the molecule stimulated cyclic GMP in cellular extract. Murad and his colleague ultimately demonstrated that acide was being converted enzymatically into nitric oxide, which served as the actual guanylyl cyclase activator. These studies also explain the action of nitroglycerin, which had been used since the 1860s to treat the pain of angina that results from an inadequate flow of blood to the heart. Nitroglycerin is metabolized to nitric oxide, which stimulates the relaxation of the smooth muscle lining the blood vessel of the heart, increasing the blood flow to the organ. The therapeutic benefits of nitroglycerin were discovered through an interesting observation. Persons with heart disease who work with nitroglycerin in Alfred Nobel's dynamite factory were found to suffer more than the pain of angina on days they weren't at work. The discovery of nitric oxide as a second messenger has also led to the development of Viagra or sildenafil. During the sexual arousal, nerve endings in the penis release nitric oxide, which causes relaxation of smooth muscle cells in the lining of penile blood vessel in engorgement of the organ with blood. As described above, nitric oxide mediates this response in smooth muscle cells by activation of the enzymes guanylyl cyclase in subsequent production of cGMP or cyclic GMP. Viagra and related drugs has no effect on the release of nitric oxide or the activation of guanylyl cyclase, but instead acts as inhibitor of cyclic GMP or cGMP phosphodiesterase, the enzyme that destroyed cGMP. Inhibition of these enzymes lead to maintained elevated levels of cGMP, which promotes the development and maintenance of an erection. Viagra is quite specific for one particular isoform of cGMP phosphodiesterase, PDE5, which is the version that acts in the penis. Another isoform of the enzyme, PDE3, plays a key role in the regulation of heart muscle contraction, but fortunately is not inhibited by the drug. Viagra was discovered when a potential angina medication has an unexpected side effects. Moving to the last subtopic of this chapter, focusing on the apoptosis or a programmed cell death. Apoptosis or programmed cell death is a normal process that is unique to animal cells. Apoptosis occurs through an orchestrated sequence of events that leads to the death of the cell. Death by apoptosis is a neat, orderly process characterized by the overall shrinkage in volume of the cell and its nucleus, the loss of adhesion to neighboring cell, the formation of blebs at the cell surface, the dissection of the chromatin in small fragments, in the rapid impungement of the curves by phagocytosis, which I will show you later. Apoptosis is often contrasted with a different type of the cell death called necrosis, which generally follows some types of physical trauma or biochemical insult. Like apoptosis, necrosis is generally considered to be a regulated and programmed process, although much less orderly in nature. Necrosis is characterized by the swelling of both the cell and its internal membranous organelles, membranous breakdown, leakage of cell contents into the medium, and the resulting induction of inflammation. Because it is a safe and orderly process, apoptosis might be compared to the controlled implosion of a building using carefully placed explosive as compared to simply blowing up the structure without concern for what happens to the flying debris. The photos are about a comparison of normal and apoptotic cells. The first picture you see is the scanning electron micrographs of a normal and apoptotic. Next image is an image of a T-cell hybridoma. The apoptotic cell exhibit many surface blebs that are bad up in the cell. In the last image is an transmission electron micrograph of an apoptotic cell treated with an inhibitor that arrests apoptosis at the membrane's living stage. To clearly understand about apoptosis, here is an image of an apoptosis curves out the structure of the mammalian digits. 3 stages in this process in a mouse embryo. In this particular mouse, which is called a Mac blue mouse, all of the embryonic macrophage express the cyan fluorescent protein. fluorescent macrophages have infiltrated the regions of the footpad where apoptosis has occurred and are clearing the space between the digits. Once a gene has been identified in one organism, such as nematode, researcher can search for homologous genes in other organisms, such as humans or other mammals. The identification of the CED3 genes in nematodes led to the discovery of a homologous family of proteins in mammals, which are now called caspases. Caspases are a distinctive group of cytosine proteases, example protease with a key cysteine residue in their catalytic site, that are activated at an early stage of apoptosis and are responsible for triggering most, if not all, of the changes observed during cell death. These caspases accomplish this feat by cleaving a select group of essential proteins. Among the targets of caspases are the following: more than a dozen protein kinases, including focal adhesion kinases, FAK, PKV, PKC, and RAP. Secondly, lamins, which make up the inner lining of the nuclear envelope. Cleavage of the lamins lead to the disassembly of the nuclear lamina and shrinkage of the nucleus. Third is the proteins of the cytoskeleton, such as those intermediate filaments, actin, tubulin, and galsulin. Cleavage and consequent inactivation of these proteins can lead to changes in cell shape. Lastly, an endonuclease called caspase-activated denase or CAD, which is activated following caspase cleavage of an inhibitory protein. Once activated, CAD translocated from the cytoplasm to the nucleus where it attacks DNA, severing it into fragments. Recent studies have focused on the events that lead to the activation of a cell suicide program. Apoptosis can be triggered by both internal stimuli such as abnormalities in the DNA and external stimuli such as certain cytokines or proteins secreted by cells of the immune system. For example, epithelial cells of the prostate become apoptotic when deprived of the male sex hormone testosterone. This is the reason why prostate cancer that has spread to other tissues is often treated with drugs and interfere with testosterone production. Studies indicate that external stimuli activated apoptosis by a signaling pathway called extrinsic pathway that is distinct from the utilized by internal stimuli, which is called intrinsic pathway. Here, we will discuss the extrinsic and intrinsic pathways separately. However, it should be noted that there is crosstalk between these pathways and that extracellular apoptotic signals can cause activation of intrinsic pathway. So first, let us discuss about the extrinsic pathway of apoptosis. So this image is a simplified model of the extrinsic receptor-mediated pathway of apoptosis. And here is the overall description happens in the extrinsic pathway of apoptosis. This image shows about the intrinsic mitochondria-mediated pathway of apoptosis. And this slide is the overall descriptions happens in intrinsic pathway of apoptosis. Another image, an image of clearance of apoptotic cells is accomplished by phagocytosis. This electron micrograph shows an apoptotic cell curves within the cytoplasm of a phagocyte. So, now, let us talk how a signal cell survives. So here it is. Just as there are signals that commit a cell to self-destruction, there are opposing signals that maintain cell survival. In fact, interaction of TNF with a TNF receptor often transmit two distinct and opposing signals into the cell interior. One is stimulating apoptosis and another stimulating cell survival. As a result, most cells that possess TNF receptor do not undergo apoptosis when treated with TNF. This was a disappointing finding because it was originally hoped that TNF could be used as an agent to kill tumor cells. Cell survival is typically mediated through the activation of a key transcription factor called NFKV, which activates the expression of genes encoding cell survival proteins. It would appear that the fate of a cell, whether survival or death, depends on a delicate balance between pro-apoptotic and anti-apoptotic signals. So that's all. That is the coverage of my report. I hope you guys learned from my report and thank you for listening. God bless.